Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Real-Time Forensic Drug Analysis Using Direct Ionization Mass Spectrometry. My name is Finn Price, and I'll be moderating today's presentation. I'm delighted to be joined by Peter Kane, Drug Scientific Advisor at Eurofins Forensic Services. Throughout the webinar, please feel free to ask questions for the Q&A session. You can submit your questions via the chat icon on the left of your screen. Without further delay, let's begin today's presentation.
Thank you very much, Finn. Hello, everyone. What I'd like to do today is to go through the work that we've done using the new Radian ASAP MS, which Walters have developed, and explain to you our use of it within a forensic drugs analysis laboratory. We're very excited by this bit of kit. It's something we've been wanting to incorporate into our laboratory for a long time, and this has been the first piece of equipment which meets our requirements. If I can first of all tell you a little bit about Eurofins. Eurofins is a leader in testing service markets throughout the world and has a strong background in forensics. The company itself has now over 48,000 staff across a network of more than 900 independent companies in over 50 countries, and we operate out of more than 800 laboratories. And you can see from the slide we have over 200,000 analytical methods at our disposal. We're split into 5 main divisions, and forensics is part of the genomics division, but we also have a food and environmental testing, a pharma and clinical diagnostics divisions. With regards to forensics itself, we are mainly based in Western Europe, in the United Kingdom, Germany, France, Netherlands and Belgium. And amongst these laboratories, we provide quite a bit of DNA profiling services. There's drugs analysis and there's forensic toxicology. And this is for police forces and other law enforcement agencies across Europe. Here in the UK, we have a large range of forensic services. Eurofins Forensic Services is the largest forensic supplier within the UK, and most of the work in the UK is actually done by private forensic companies. I'll talk about that a little bit further on though. Our services include, as well as the drugs of abuse and toxicology, we have biology services, DNA, firearms, marks traces, sexual offences work, cold case reviews, we do coroner's toxicology, digital crime and old-fashioned question documents, handwriting experts as well. Now I'd like to tell you a little bit about the drugs teams at Eurofins. We can trace our origins back to the 1920s when we were part of the laboratory of the government chemist, where we first started to analyse opium and cannabis that was seized in the London opium dens. Since then, you can see from this slide how we've grown from a team of a couple of analysts originally to currently having about 70 staff across 4 different laboratories within the United Kingdom. Within the UK, we work to 3 main pieces of legislation: the Misuse of Drugs Act 1971, the Misuse of Drugs Regulations 2001, and the Psychoactive Substances Act 2016. This slide I'm showing you at the moment is a simplified version of the Misuse of Drugs Act 1971, and as you can see from it, apart from named drugs, we also have what we call generic controls, and these are essentially paragraphs of chemical text which also indicate which other drugs, apart from the named ones, are controlled. We also have the 3 classes of drugs, class A, B and C, and if there's ever emergency legislation on a particular compound required, we have what's called a temporary class drug order. Most of the main big drugs, such as cocaine and heroin, obviously fall under our Class A, but other ones which are deemed not quite as harmful in the UK, such as amphetamine sulfate or cannabis products, fall under Class B. This bit of legislation, even though it doesn't look it from that slide, covers something in the region of 5 million different drug compounds, and it's the job of myself and my colleagues to identify them whenever we come across them. This next slide actually shows our workflow. So this is how we were doing things before we got hold of the ASAP MS. We unwrap all of our packaging. We get actually given the whole sample. So if it's seized as a packaged block, then we have to unwrap it. We don't just get a subsample, we get the whole thing. We therefore then weigh if it's a powder, if it's tablets, we count the number of tablets present. If it's a liquid, we'll do the volume. We then come to our screening techniques, and these are a mixture of either colour tests, thin layer chromatography, or FTIR. And we can also use characteristic identifiers such as tablet markings, or if particular packaging or labelling gives us an indication of what might be present. We'll then confirm that by using either FTIR or GCMS. There are two routine go-to techniques. However, if necessary, if we come across something new which we haven't seen before, we do have access to both NMR and high resolution accurate mass LCMS. We only use those when absolutely necessary. We also undertake purity determinations in the UK, and we do this by HPLC mainly, GCMS for some samples, and we use GCFID for cannabis products. In the UK, our sentencing can quite often be determined by the purity of the drug and the weight of the drug. And we have police experts who will work out what the street value of the drugs are, and they put that forward as part of their evidence, and it can be used towards a sentencing if the defendant's found guilty. As I mentioned earlier, we see the whole sample, not just a small amount of it. Eurofins drugs teams work to the SWIG drug guidelines, and SWIG drug is a scientific working group for the analysis of seized drugs, and their website is swigdrug.com. We are regulated by the United Kingdom's Forensic Regulators Office's Code of Practice, and every one of our scientists has to sign up to the regulator's Code of Conduct. It's essentially a list of 13 points that we agree to as forensic scientists that we will work to undertake our work properly, correctly, and without reproach. All of our work is accredited by the United Kingdom Accreditation Service to ISO 17025 as a testing laboratory, and all of our work is reported on what we refer to as streamlined forensic reports. So that essentially gives the weight, identification, purity of the drug found. If, however, this is challenged by the defence, or we're required to give evidence in court, we have to then do what we refer to as a full witness statement, and in that instance that can be used in the court of law with one of our scientists present. So where does the ASAP MS come into this? Well, part of my remit as the drug scientific advisor is to look at new bits of instrumentation or emerging techniques that are on the market or being produced to see if anything can help with our work, what can make us quicker, what can make us more efficient. At the moment, within drugs, and as you saw from the diagram earlier of the Misuse of Drugs Act, there are millions of compounds that we have to test for. And as a consequence, there is only a limitation in the number of colour tests or TLC screening tests that are available to us. As a consequence, not all of the drugs that we see react to colour tests, main group being the synthetic cannabinoids or spice compounds. Our TLC systems can be a bit slow. They can take between 40 minutes and an hour, some of them, depending on the mobile phases used with them. And in some of our laboratories, they actually don't have spare fume hood capacity to undertake TLC, so they have to put all of their samples onto GCMS. And this, unfortunately, leads to bottlenecks, and we can end up running a lot of samples for one case in order to meet our sampling requirements. A long-term goal of mine has always been to find a technique that required very little sampling preparation and could be used for those drugs that our existing screening methods don't work for. As a consequence of looking for a new technique, I was in discussion with colleagues from the National Measurement Institute at LGC, which shares a building with us, and they indicated that they might have a new instrument which Walters were developing, which they believed might be of use and which they were testing at the time. Within the United Kingdom, cost is a big factor in forensic analysis because we're all private forensic suppliers and the police forces pay for our services. And in the UK, the police also have limited budgets. They want prices to be kept as reasonable as possible, and therefore, as a forensic supplier, we want to keep costs as low as possible. So this brings us to the radio instrument, which is Rapid Direct Analysis Atmospheric Pressure Sample Analysis Probe. It was specifically designed for rapid, easy, low-cost sample analysis of solids and liquids. And we found, yes, it works for a lot of compounds. So what's the ASAP ionization technique? So we introduce a sample on the end of a small glass rod. The sample preparation is very simple. We dissolve our samples in methanol, dip the end of the glass rod a couple of millimeters into the liquid, take it out, give it a few seconds, then it can be introduced into the instrument. The sample on the end of the rod is introduced into the corona discharge region. It's then volatilized by a stream of heated nitrogen gas. The gaseous analyte molecules are then ionized by the nitrogen plasma, and the gaseous ions are guided into the instrument and then analyzed by the mass spec. The next slide shows the ASAP mechanism of ionization, and I'll admit that I'm not an expert in what's going on in these slides, but I can describe it the best I possibly can. The first is a charge transfer mechanism, which is initiated by the corona discharge ionization of the nitrogen in the source, which generates radical cations of nitrogen, which can then undergo charge transfer with analyte molecules to generate radical cations of the analyte molecules. This form of ionization is particularly useful for the analysis of non-polar compounds. The second mechanism for ionization is by proton transfer, and this requires the presence of water or other solvent which can act as a source of protons to form ionized solvent clusters, as shown. We were using methanol for this part of the proton transfer. The ionization of the analyte then occurs by proton transfer, which is the main ionization pathway associated with APCI in LCMS work. So let's have a look at the mass spec screening approach. The fragmentation is produced by in-source collision-induced dissociation. The higher the energy we apply to the sampling cone through which the ion enters into the mass spectrometer, the higher the degree of fragmentation that occurs. The ASAP ionization process produces mainly intact ions with very little fragmentation, which is unlike EI ionization in GCMS, which does result in a lot of fragments. So to increase in specificity, the cone voltage fragmentation is used to produce characteristic fragments. The higher the cone voltage, the greater the fragmentation. Increasing the cone voltage increases the energy of the ion entering the mass spectrometer. Here on this slide you can see we've got ecstasy, which has a mass to charge ratio of 194, and this is with the M plus H, because ecstasy itself is 193 in its normal state. The instrument, because it uses a softer ionization than GCMS, we can determine the cone voltages, and the instrument itself allows for 4 different voltages. So we've been using 15, 30, 50 and 70 volts. And this gives the additional specificity for the identification of by the generation of the fragments. The next slide actually shows how the fragmentation works. On the right-hand side, the spectrum showing is from the lower voltage at the bottom, and increasing upwards. You can see from the bottom spectra that the 194 ion is quite dominant. As you move up the spectrum, that one slowly disappears and other ions appear. And this is quite characteristic for ecstasy. Where we've been working with Walters, we have put together a library, and at the moment has about 50 common drugs, and they're mainly the ones that we've been seeing most often within our laboratory. On average, we see around 350 to 400 different drugs a year, so we decided just to pick the top 50. Walters have developed LiveID software, which is the library matching program that we've been using on this work. And it can be used in both real time, and I'll show you an example as we go forward, or for matching pre-acquired files. We've used both, and we found them both to be very useful moving forward. Their algorithm matches acquired on the spectral data, and the spectral matches are performed on the 4 different voltages, which will give you a result up to 1,000. These 4 different voltage matches are then averaged, and the average calculated, and that gives you your match criteria. In this slide, you can see an example of the total ion current, which is shown, and that cocaine has been identified. This slide shows data from 3 replicate samples analysis. Data can be viewed from each replicate. In the upper portion of the slide is the TIC, and the lower portion of the slide shows the spectra from sample and corresponding library. The library works by picking 5 ions at each voltage and then comparing each of those 4 sets of 5 ions against your unknown, and the spectral match is made accordingly. The system can also accommodate mixtures. In this case, 2 similar amphetamines are identified, being MDMA and MDEA. Slide, you can see this one is a mixture of MDMA and MDA, again 2 amphetamine compounds. This next slide, which is using the MassLink software, shows 3 Xanax tablets which were run in quick succession. These particular tablets each contain alprazolam, but we know they're actually illicit tablets, as we've seen these on a number of occasions within the UK. You can see from the retention time beneath each of the peaks just how quickly you can load up a sample and get a result out. So from our perspective, the speed of this technique has been very beneficial. The next slide I'm showing is of heroin powder. Within the UK, the heroin that we normally see is a brown colour, and it contains at least 5 components, these being the diamorphine itself, 6-monoacetylmorphine, 6-acetylcodeine, noscapine and papaverine. This particular powder is at 70% pure, which is quite strong for a UK heroin sample. And from this you can see that the 370 iron is the most dominant one, showing a good match for heroin. However, this next slide shows a heroin sample which is at 20%, and this particular one is cut with caffeine and paracetamol. The spectra you can see though also shows some of the other opiates which I've already mentioned, such as the noscapine, but we can actually pick out the different ions from the caffeine, the paracetamol, the noscapine and the heroin itself, which is very good, seeing as in the UK a lot of our samples are impure or heavily cut. So they've either been adulterated at source or they've been cut before they've been sold on the streets in the UK. To give you an idea now of where we believe the ASAP MS will save us significantly is when we currently examine herbal substances which we believe to have either synthetic cannabinoids or more also known as spice compounds. The only way we can analyse these at the moment is by putting them through our GCMS instrumentation twice, almost one as a screen and one as a confirmation. Because under SWIG drug rules you have to test, or should test, 2 independent samples of your unknown. And the only way we can do this at the moment is by using GCMS twice. As you can see from the slide, including our blanks that we have to run in between every sample, a typical sample can take us up to 50 minutes for a synthetic cannabinoid. And if we have more than one package present, that does increase by, it can be 5 minutes, it can be significantly longer depending on the number of packages present. However, using the ASAP MS actually allowed a lot quicker screening to be done. We put samples onto the instrument and within a couple of minutes we had all of our results off. And what the technique also showed us very easily, which was something we were very pleased with, is if you've got different synthetic cannabinoids within different packages, it was easy to actually pick out which bag had which synthetic cannabinoid on without having to do the full GCMS run. And this is shown in the next slide here. And you can see that with the 3 samples, that sample 2 and 3 are the same, whereas sample 1 is a significantly different synthetic cannabinoid. When we actually did this experiment, we actually used one of our technicians who's not a trained drugs analyst, and we asked them to run these samples just to see how simple the process was, and they managed to analyse these samples with no problems at all. It is a very easy press and go system, something that, as I say, we're very grateful for in a high throughput forensic laboratory. So now we have this equipment in, how are we going to use it? I've put now up our workflow moving forward. And you can see that under our screening is where the ASAP-MS will be of benefit, but also there will be instances when we have purer compounds where we can do a colour test, say for ketamine, for example, which doesn't have many other compounds which react with the colour change, and then we could put that onto the ASAP-MS, and that will satisfy the SWG drug criteria. So in that instance, we can get a result out very, very quickly indeed. Ketamine in the UK is very rarely cut, so again, there shouldn't be any interferences which would stop us from being able to identify the compounds. We are currently undertaking validation of the instrument, and we hope to get it into our accredited method list very, very soon. Most of the work has been done, we're now just finishing off, but it does look as if it will be able to save us significant amounts of time through screening and on occasions confirmation work. So what are some of the advantages we've seen? The analysis in real time, so results are obtained within minutes of sample preparation, has been great, especially for unknowns. Multiple samples can be run in a sequence using the live ID, so when we do have multiple units, we can screen them. We're getting positive mass spec identification, and we can run a large number of samples one after the other. The preparation and runtime has been reduced by at least 50% compared to our thin layer chromatography analysis. On average, it takes us about 40 minutes to do 14 samples. With the ASAP-MS, we've got that down to under 20. So significant time savings have been seen. As could be seen with the previous slide, we now want to use the ASAP-MS with other techniques, and it can be regarded as either a category A or a category B technique under the SWG drug guidelines, depending on the sample being analysed and how pure that sample is. Sample preparation is minimal, it's literally a dilution. Occasionally we'll filter the solutions as well if we think they're too cloudy or they've been made too strong. And our target compounds can be detected in street samples down to at least 10% purity, which for the heroin samples, which I showed you earlier, is a great benefit. There have been some challenges. It hasn't all been plain sailing. The instrument does run on nitrogen, so you will either require a pipe gas supply or a nitrogen generator. Originally, we were hoping that this would be a portable piece of equipment, and discussions with Walters engineers, they were hoping it could be portable too, but the current Radian model isn't as portable as we'd hoped. However, that's a minor thing at this stage. We're hoping that in the future they can make it into a more portable model. The instrument was planned to be used in some police stations, as Eurofins supply scientists into some police stations to help work if there's a large festival on or something where they believe submissions could increase. Unfortunately, the police stations won't allow piped gases to be used, and as a consequence we therefore can't use the instrument in those settings. Originally we were in discussions that air might be able to be used as the carrier gas within the ASAP-MS. However, that's something that Walters will be looking into in the future. It would be a great benefit for us if they could do that, but at the moment we're not going to be able to use this bit of equipment in police stations. As with GC-MS limitations, we can't examine different isomers, and in the UK we do see a lot of drugs which are isomers of each other, methyl methcathinone, for example, we do see the 2, 3 and 4 isomers, and the only way we can really distinguish them easily is by FTIR. So in such instances, the ASAP-MS is not going to be the instrument of choice. The spectral libraries will need constant revision. In the UK, the drugs market is constantly changing, and we do regularly see new compounds. And these can be from the synthetic cannabinoids to cathinones to amphetamine derivatives. And as such, we would have to include these in any libraries moving forward. However, the 50 compounds that we have put into the library so far do cover the majority of the routine samples we see. The software, the Live ID software, is bespoke for this instrument, and I take my hat off to the Walters software engineers because they listened to us. We explained what we required from the software, and they worked on it, and they have given us a product which is easy to use and gives us exactly what we want. The original software we were using wasn't really fit for purpose and did cause some issues, but the Walters team have responded very well and written a new software which now works extremely well for our purposes. So to summarise, the ASAP-MS has shown it has the capability to screen samples not currently able to be done with our current tests that we use or the ones that require lengthy screening methods. It gives us a rapid real-time testing on minimal sample amounts and requires minimal sample preparation. It's an easy-to-use sample introduction technique, and batches of samples can be run in approximately 50% of the time that our other current methods take. I'd just like to make some acknowledgements at this point. My colleagues at Eurofins, Ryan Francis and Kirtan Patel, have worked over the last few months on the equipment, and they have been full of praise of its capabilities, and I pass my thanks on to all their hard work. My colleagues in the National Measurement Institute at LGC, Chris Hopley and Brian McCulloch, who are both experts in mass spec, and they have been invaluable in helping us when we've come across different issues. And also at the Walters Corporation, Dr. David Doose and Dr. Nicola Lumley for their help and support whenever we've required it. We also have some further reading for you if you are interested. A number of us put together a paper which has been published in the Journal of American Society of Mass Spectrometry, and the details are there on the slide should you want to read a bit more on the work that we've done. Thank you.
Thank you very much, Peter. Just before we move on to the Q&A session, I'd like to bring everyone's attention to some poll questions that we're just about to push out. We'd be very grateful if you could give us your answers to these. So then let's go straight on to our first question today, and that is, what are the major changes in the types and amounts of drug seizures that you have seen over the last 10 years? Sorry, everyone. We seem to have briefly lost our presenter, Peter. If you give us just a couple of minutes, we're just trying to get him back on the line.
Hi, Finn. Sorry, can you hear me now?
Yes, we've got you. Shall I repeat the question for you?
Yes, please. Sorry about that.
Not a problem at all. So the question that we had was, what are the major changes in the types and amounts of drug seizures that you have seen over the last 10 years?
Major difference we've seen has been a move towards more synthetic drugs. Cathinones, for example, have really shot up the number of submissions we have of those, and the synthetic cannabinoids as well have been a definite change, the number of samples of those we're seeing. On average, we probably see about 10 to 15 new drugs each year, which we haven't encountered before. And they do tend to be samples that have been seen around the world, but occasionally we will get something that can be a challenge, shall we say, and that's where we'll employ the NMR or accurate mass techniques to help us identify them. But yes, things that have gone from sort of mainly being cannabis, heroin, cocaine hydrochloride to a large number of these synthetic drugs. Okay.
Thanks very much. Next up we have, how easy was the system to use for the operators at Eurofins?
As I mentioned during the presentation, we found it extremely easy to use. The sample preparation was quite minimal. Literally dissolve a small amount into a little bit of methanol, dip your glass rod into it, give it a little shake just to take off the excess liquid, and introduce it into the instrument. It's very, very simple bit of kit.
What was the typical sample preparation that you used for seized drug samples with the Radian ASOP system?
As mentioned, it was really, we would grind the sample to make sure it's homogeneous, take a small amount, dissolve in some methanol, and introduce it into the instrument. And so, as I say, we could do that with virtually every sample we had. Only issues were when you get something like a steroid, which might be in an oil-based solution, that could cause some problems, so we tended not to introduce those sort of samples.
What improvements would you like to see in future developments of the system?
One thing, and I did mention it during the slides, is that the instrument does use nitrogen. Now, when we were with the scientists from the National Measurement Institute, they had been using a research model using air, and it was working very well, and we ran a large number of our samples just using air as well. So if that could be introduced in the future, that would be great for us. As I mentioned, it would then take away some health and safety risks with regards to using just air as a gas as opposed to nitrogen or something else, which would require either a generator or piped gases.
Are you seeing increasing sample requests for novel psychoactive substances, or is it just the kind of more common illicit drugs that you're mainly asked to identify?
Well, our main drugs such as heroin, cannabis, cocaine, they still probably account for about 85% of the samples we see, but we are definitely seeing an increase in the synthetic drugs, especially the cathinones and the synthetic cannabinoids. We have seen some of the new fentanyls as well, which I'm aware of the US has seen quite a number of. However, they've started to disappear again, so we're hoping that that was just a small episode with regards to fentanyls, and we're hoping it's left the UK again at the moment.
Our next question is just to clarify around the use of the ASAP system a little bit. Is the operator required to be present during the analysis, or can samples be pre-prepared and some sort of auto-sampler set up?
The Radian that we have at the moment is a manual system. You can pre-prepare your samples as long as you've got them ready in a small vial, already in dilution. But it's actually a manual introduction technique at the moment. I know Walters have discussed this with us, maybe something in the future that they'll go on to making some form of auto-sampler for it, but at the moment it's a manual introduction technique.
What is your experience with carryover when dealing with highly concentrated or highly pure seized drugs?
Well, it's very easy to actually see carryover. The slides where we actually showed you the Xanax tablets, where we analysed them quite quickly, one after the other, you can actually see the baseline returning to its lowest point. So you can see if you have a carryover there. So yes, occasionally we did overload, and I mentioned about steroids being in oils. They were something that particularly caused that. But you could see in live time whether or not it had gone back down to its baseline. So from that perspective, carryover was fairly minimal.
Can the instrument be overloaded at all? And again, linking back to last question, is carryover a problem at all?
You can overload any instrument, but it was very quick that it could come back down. There's minimal amount of parts that you can, as a user, get to, and the cone part of the ASAP part of the instrument is the only bit that we ever had a problem with, and we changed that once during the 6-7 months that we were trialling the first instrument that we were using. So it's difficult to overload it, but you probably could if you really wanted to try.
Great, thank you very much. How does your approach address the identification of positional isomers and diastereomers?
Well, as I mentioned during the talk, that as with other mass spec instrumentation, it's not always possible to see different isomers. You can in some instances, but not all the time. So we tend to use FTIR in those instances because it's much better at identifying different isomers and stereoisomers.
Have you evaluated any other direct introduction techniques?
No, this was the first one we had because, as I say, my colleagues in the National Measurement Institute were trialling it on some other research and development projects, and we were fortunate that Walters were willing to work with us to see whether or not the ASAP instrument would work in our environment, and it did. But no, we haven't tried any others, not because we didn't want to. It's just the opportunity arose to use this Walters one, and we used it and found it to be exactly what we were after.
Did you do the quantification of drugs using this instrument?
No, the software currently with it isn't for quantification purposes. We could possibly have tried, but that wasn't our intention. It wasn't what we were hoping to use the instrument for. But again, Walters are aware it is a possibility, something for the future, and if they can get the software written, then I don't see why it couldn't be used as a quantitative technique as well as qualitative.
Pardon me, sorry, I just had my microphone on mute there. We've got one last question that we can fit in, I think, and that is, what is the instrument's sensitivity in terms of micrograms or nanograms per sample?
We went down to, we were using a milligram per mil as our general rule of thumb, and we could probably go down to about 20 micrograms per mil and still get reasonable peaks out.
Great, thank you very much. Thanks for answering so many questions, and thank you all in the audience for sending so many in to us to ask as well. Unfortunately, that's all we have time for today. It's been a great presentation, and yeah, thank you all for joining us. If you have any other questions, please feel free to email me at editor@selectscience.net, and I'll follow up with these personally. If we've not been able to answer your question either, we'll follow up with these too. Remember, you can download a certificate of attendance from the Documents tab on the left of your screen. If you'd like to listen to the webinar again or invite a friend to listen, they'll be available to watch on demand in a few days' time. Goodbye, and thanks once again for joining us.